So, hi everyone, my name is Lorenzo Masoero, and today I'm going to talk to you about exciting work that I've been doing with my advisor, Tamara Broderick, as well as co-author Josh Schraiber. And so the main theme that we're going to consider today is that although there is a ton of genomics data, many scientists are still limited by a fixed and finite budget when designing their genomics experiments. So my work shows how they can learn the most possible information given a fixed and finite budget, especially in the context of rare variance association studies. So to give a little bit of an overview of the problem that we're going to consider today, let me just start by saying that the problem discovering genomic variants, including rare genomic variants, is of extreme interest in the scientific community. For example, in human genomics, variants are useful for understanding disease such as cancer and for precision medicine and effective targeted drug design. So what's happened is that in the last 2 decades, next generation sequencing has provided larger genomic libraries and higher quality samples. And these two factors combined have allowed scientists to start uncovering the genetic base of disease using studies of common variants like GWAS. GWAS, these genome-wide association studies, and more generally studies on common variants, on the one hand, have had great success and played a fundamental part in explaining the role of common variants in disease. But on the other hand, however, there is a large fraction of unexplained heritability that is not due to common variants and is instead due to rare variants. So to uncover this missing heritability, we then need association studies and related statistical tests that are specifically designed to investigate the impact of rare variants in disease. And these are the studies that we will focus on today. While studying these rare variants sounds appealing in principle, there are a number of limitations that we need to overcome in order to effectively study the role and impact of rare variants on disease. So fundamentally, rare variants association studies are costly and hard to design. And the reason is that infrequent variants cannot be studied individually. We need to collect and aggregate many rare variants in order for our statistical tests to be informative and conclusive about association of rare variants with disease. Then, to get more rare variants, we could sequence more individuals because a larger genetic pool would ensure to observe more rare variants. Or alternatively, we could increase the sequencing depth of the sample, that is, design sequencing schemes that allow us to minimize the number of variants that are missed in each sample. The problem is that both options are expensive and result in a higher cost of the experiment. And since experiments are always conducted under a fixed and finite budget, it will be crucial for us to identify the best choice for these parameters in the service of maximizing the usefulness of the experiment. So our proposal today will be to provide a framework for the optimal budget allocation, that is choosing between the sample sizes and sequencing depth of the experiment, with the goal of maximizing the power of a statistical test for rare variants that we will conduct on the collected sample. So just to give a little bit of a biological background, in modern genomic data sets, we observe individual genomic sequences. And here's a sketch of one such sequence. So the atomic unit of the sequence is a base or nucleotide, and fixed positions on the sequence identifying genes make up genomic loci. And different loci, different sequences can have a specific expression or alleles for the same locus. So another input of the problem is a standardized ideal genomic sequence for the population under study, the so-called reference genome. So for every sequence and for every locus, we can compare the expression of that locus to the one of the reference and create a binary encoding of variation. So here, for example, the expression on the first locus coincides for both sequences, and the same is true for the second locus. By the third locus, you can see differs. So we can keep on performing this comparison for every locus and create a map of variation. We say that a variant is called whenever the allele differs from the reference. Pictorially, we encode this information in a binary vector in which every square represents a locus, and the square is black whenever variation is observed. If we perform this comparison for every observation for every genomic locus, we end up with a sparse binary matrix in which the rows are the individuals and the columns the genomic loci. But for this talk, rather than considering this matrix, we're going to consider a permuted version of it, the so-called left-ordered form, in which columns are sorted by order of appearance or a fixed ordering of the rows. In this way, we destroy all the spatial information about the genome, but we see immediately that the columns quantify a rate of growth of the number of observed variants. So in a nutshell, we will next design a statistical model that will allow us to accurately predict the number of new variants observed in a new sample as a function of both the size of the sample as well as the experimental conditions under which the experiment is performed. as quantified by the sequencing depth. And so far I've mentioned the sequencing depth a number of times, but I've not explained exactly how this quantity impacts our experiment. And so the idea is that collecting genomic samples is a complex, expensive, noisy process in which the final output depends on a large number of parameters that experimenters can tweak and modify. And one crucial aspect of the sampling process is actually the choice of the sequencing depth or coverage. So we'll denote the sequencing depth by lambda, and intuitively, this is just a scalar parameter that quantifies how accurate the measurements made are. So a higher depth implies higher quality samples, but also more cost and use of resources for every single observation. A lower depth, vice versa, will lead to coarser and noisier measurements, but will be cheaper. So under a fixed budget, we will want to maximize the power of a statistical test of the data, and we will have to do so through optimal experimental design, trading off the quality length of the experiments with the quantity M of samples that we will be allowed to collect under a fixed budget. And so just to give you a little bit of a taste of what these rare variance association hypothesis tests look like, fundamental idea is that we want to be able to answer a question: are rare alleles associated with disease? So to do so, what we do mechanically is to first collect data from the two populations under study, so an unaffected population and an affected population. And next, we compare fundamentally the abundance of certain rare variants in both populations. So here we will focus on singletons burden tests, that is, statistical tests that count the number of variants appearing exactly once, that is singletons, that is pictorially the number of columns with only one non-zero row. And we'll do so by quantifying the average number of singletons per patient in a population J, where J is either unaffected or affected. And so the test that we will perform will be to ask ourselves, is the expected number of singletons equal in both populations, the unaffected and the affected population, or is it instead true that the expected number of singletons in the unaffected population is smaller than the number of singletons in the affected population? And so to test the null hypothesis of basically absence of impact of rare variants, we'll define a statistical test. So defining NJ to be the number of data points in population J, where J is either unaffected or affected, and mu bar J, the average number of observed singletons in the population with related standard deviation Sj. We'll compute this test from the data, mu bar A minus mu bar U divided by a scale version of the pooled standard deviation. And here, the crucial aspect to understand it is that both mu bar J and Sj jointly depend on the sample sizes that we've collected, n sub A and n sub U, and the sequencing depth of the experiment. So the goal really to allocate our budget optimally so as to maximize the power of a test for a given target confidence level. That is, this is the quantity that we will want to optimize. So the probability of rejecting H0 given that H1 is true. And to tackle this problem and address the previous question, we adopt a Bayesian nonparametric approach. So Bayesian nonparametric models are known to grow in complexity as more data is collected. And because we're thinking of variance as a feature of the population that keeps on being uncovered from the population, a BNP model seems like a natural choice. Moreover, the IBP, the Indian buffet process, is a standard tool within the BNP framework. It's fundamentally a prior for equivalence classes of left-ordered form binary matrices. That is, a sample from the IBP looks like something like this. On the rows, we have the samples, and on the columns, the distinct variants in left-ordered form. So that if we invert the axis and count the number of variants on the y-axis as a function of the number of samples, we immediately see that for every row, this induces an ever-increasing number of variants, just like we expect to see in the data. So our approach will be fundamentally to try to fit this curve and then use this fit to inform the statistical test and the optimization problem. And there's a number of issues we have to overcome. First of all, the fact that the IBP is a stationary model that is given collected data. Maybe we want to gather more data under different sequencing depths. The IBP would not accommodate that. So to overcome this, we leverage a recent model we developed for another publication that allows us to model basically data generation under changing sequencing depth. Moreover, here we really have this affected, unaffected population that we need to model jointly, but existing models only consider one population at a time. So we developed a new hierarchical model that jointly considers affected and unaffected populations. And so then what we will do is we'll try to optimally design data sequencing to maximize the power of the burden test data that was defined before. That is, we'll try to maximize with respect to the variables that we can control, that is the sample size n sub a, n sub u, and the sequencing depth lambda, our objective function, that is the probability of rejecting the null hypothesis given that the alternative is true, subject to a budget constraint, that is the cost of the sequencing experiment cannot overcome and exceed the budget. And again, NU is the number of unaffected samples, and sub A is the number of affected samples, and lambda is the sequencing depth. That are all parameters that we imagine we can control. And so just to give a taste of what we can provide, I will briefly discuss our results here. And again, I'm going to focus on burden tests of variance, of singletons actually. So first, we consider the fixed design regime, where we ask ourselves basically how many more samples are needed from the data in order to achieve a target level of power in a statistical test at a fixed sequencing depth. So here we are imagining to keep the sequencing depth fixed. And for example, this is one sequencing depth that I'm plotting. So on the vertical axis, we have the power of the test. And on the horizontal axis, a number of samples. So we see that as we increase the number of samples jointly at the same rate in the affected and unaffected population, if we impose H1 to be true in the data, as we did, the power of the associated test increases as we increase the size of the sample. And this is natural because we are observing more evidence that the the alternative is true. So for example, if the target level of power that we want to achieve is 80% for fixed confidence rate, we see that at this fixed sequencing depth, we're going to achieve it at, for example, n equal 180 samples. But if we vary the sequencing depth, this rate changes. For example, if we increase up to lambda equal 40, we'll see that fewer samples will suffice to achieve the same sequencing power of the test. Vice versa, if we reduce the sequencing depth, this can dramatically take longer. Another question that we address is the one that I've hinted at before, that is the fixed budget scenario, in which the question we answer is, what's the optimal sequencing depth? Is the depth maximizing the power of a statistical test under a fixed budget? So here again, on the vertical axis, we have power. On the horizontal axis, we now have the depth of the experiment under a fixed budget B. So for this fixed budget, we see that basically collecting samples at a very low depth is not very informative because we're going to miss too many of these rare variants. So the usefulness of the experiment, that is quantified by the power, increases up to, let's say, a point, after which basically further increasing the depth is not beneficial because it will make us incur in too small sample sizes. So even though for every single sample we're going to observe many more variants, overall we're going to be missing out on variants because the sample sizes will shrink too much. And again, we see that if we change the budget available here by this different color scheme, the results will change. Namely, as we increase the budget, that's always beneficial. So for the same level of depth, we always achieve a higher power. And of course, we can play with these parameters and with the data generating process, and on different data, the results will change. But the fundamental point is that these curves will have nice interpretations. So this will always be monotonically increasing, and this will typically have only one clear optimum. Okay. So just to wrap up and provide a little bit of a conclusions, what I presented today is a new method that we developed to apply our Bayesian nonparametric framework for prediction and design in genomic studies. And so here we focused on optimizing the sequencing depth for power trade-offs in rare variance dissociation tests. So you can find this in our submissions to the LRML workshop. And this is still ongoing work, so any question or feedback is very appreciated. And there's also next steps that I have not discussed yet. So first of all, what I've shown here is not really our ultimate goal, that is power maximization analysis in follow-up studies, that is conditionally on data that we've already collected. How do we maximize the power for future data that we're going to collect? And moreover, this is actually on synthetic data where we had control over enforcing the null versus the alternative hypothesis, but it would be exciting as a needed next step to apply actually this model to real data and see what results it provides. And if you're interested in additional resource, references, you can look up our More for Less Predicting and Maximizing Genomic Variance Discovery via Bayesian nonparametrics that is soon to appear in Biometrica, but you can also find it on arXiv that provides a little bit of a statistical foundation of our method, as well as other work on arXiv that provides extensions and additional feedback on the hierarchical construction of our model. I'd be happy to take any questions, suggestions, or feedback, and thanks for your attention.